All right, cuties, welcome back to the second half of photosynthesis. Hopefully you remember what we have been talking about. We had been talking about the light reactions. So remember you had photosystem 2 that ejects its electrons. It goes through an electron transport chain. All this is happening in the thylakoid. Hydrogens are getting pumped into the thylakoid space. And then those hydrogens are being used to make a small amount of ATP. Then in photosystem 1, a photon hits it, its electrons are ejected and eventually caught by NADP plus, converts it into NADPH, which is an electron shuttle. And now you can see on this diagram here, the ATP that just got made and the NADPH are going to be transferred to the Calvin cycle. And carbon dioxide, you can see from this image right here, carbon dioxide is being brought in as well. All right, so the Calvin cycle is said to occur in 3 phases, and you are welcome to just memorize these 3 phases. If you remember from the Krebs cycle, or the also called the citric acid cycle from cell respiration, it is phenomenally complicated. If you look at this particular diagram, each one of these arrows represents another molecule that's being made and another enzyme that's rearranging, and oh my gosh, it's just huge. You don't need to know all of that information. Basic things, let's see if I can think of just the ultimate what you need to know. You need to know that total 6 carbon dioxides have to come in order to make carbon dioxide. So each oh my gosh, I said that wrong. 6 carbon dioxides need to come in order to make glucose, because glucose is C6H12O6, and each molecule of carbon dioxide only has 1 carbon in it. So we're going to actually take about 3 carbon dioxides in at a time, and then this cycle is going to produce a molecule that has 3 carbons in it, and then that molecule is going to get put together to make glucose. So the first stage is called carbon fixation, and usually when we're talking about fixing something, we're talking about bringing it out of the atmosphere and putting it into like an organic molecule. So you can talk about nitrogen fixation. There are bacteria that pull nitrogen out of the atmosphere and then turn it into an organic molecule. This is carbon fixation. We're pulling carbon through the stomata, remember, on the bottom of the leaf, bringing in carbon dioxide, and we're going to add it to molecules, and we're going to join them together. This is an anabolic process that requires the sun's energy in order to occur. It doesn't occur naturally. It just doesn't occur naturally. Okay, so carbon fixation, and then there's specifically, this is an important enzyme that's called ribisco. It stands for ribulose bisphosphate carboxylase, but everybody just abbreviates it ribisco. Ribisco is an incredibly important protein/enzyme that's in charge of collecting the carbon dioxide, and it's going to come back in just a minute. So we need to know ribisco. Now, reduction is what's going to be happening next. And remember, reduction is when we start collecting electrons. So ATP and NADPH are used to rearrange RUBP, ribulose bisphosphate, into PGAL. Oh my gosh, these words, I'm sorry. So glyceraldehyde 3-phosphate, also called G3P, also called PGAL, which is a 3-carbon sugar. So what you need to understand, carbon dioxide comes in and it gets fixed. That's called carbon fixation by ribisco. You need to understand that 3 of them get put into a molecule of PGAL. And then you need to understand that it gets recycled. All of this is getting recycled. So the wheel is turning repeatedly. And every time 3 carbons are leaving the system, 3 more carbons are entering the system, so that the system can keep rotating around and around. But other than that, you don't need to know any of the painful details. All right, so, and I forgot to say this, the question over on the side here, where's the sugar? Where is glucose being made? Because I mentioned that we make this molecule that has 3 carbons in it called PGAL. Well, then the cell is able to put 2 of those 3 molecules together to make your C6H12O6. So in order to get 1 G3P, or also called PGAL, as a product of the Calvin cycle, 3 molecules of carbon dioxide have to be joined to 3 molecules of RUBP. This makes 6 molecules of G3P one of which is a net product. So P-gal, as it's turning, one P-gal leaves, 3 carbon dioxides have to come in each time. So the other five G3P molecules are used to regenerate 3 molecules of the RUBP. G3P is a sugar building block. 2 of them can make 1 6-carbon sugar. Many of them make a polysaccharide. Calvin cycle, named for the dude who figured it out. It's also simply referred to as carbon fixation, never called dark reactions. So don't refer to them as the dark reactions. Okay, and then inputs and outputs. So what's coming in total? So we're talking about net. 3 carbon dioxides are coming in, and we're releasing one of these P-gal, one of these G3P. It has 3 carbons in it. We are also taking in 9 ATP and taking in 9 NADPH. We're removing electrons, so they are getting, we're adding electrons. I got to think that through really quickly. We are removing electrons. Oh my gosh, that was a little confusing for me. And we're ending up with ADP plus inorganic phosphate and NADP plus. All right, and then, so that's all of photosynthesis right there, but there's some other details that we're going to want to know about. So versatility and regulation. It turns out that our ribulose bisphosphate, Rubisco, this super important enzyme, has something that's a little bit wrong with it. Rubisco is believed to have evolved in conditions that had low oxygen gas concentration. As a result, its active site has a high affinity for oxygen gas. So what they're saying is that it didn't matter that Rubisco likes oxygen because there was never very much oxygen available when it first evolved. But now there is a lot of oxygen available, and so now it has a tendency to click onto oxygen. It's supposed to be attracted to carbon dioxide. That's what we're pulling out of the atmosphere is carbon dioxide, but it can also attract oxygen, and that's not what the plant wants it to do. So that can be a problem. So if the Rubisco, instead of attracting carbon dioxide, starts attracting oxygen, that's called photorespiration. And then it says here, the metabolic pathway that occurs when Rubisco incorporates oxygen instead of carbon dioxide. It is a metabolic dead end. It uses ATP, but it does not produce any sugar, and this is bad for the plant. The plant does not want this to happen. So there are all kinds of things that have happened in order to prevent this from happening. So let's move on. So lots of times it's not a problem. As long as the plant keeps its stomates open and exchanging gas with the environment, photorespiration is kept to a minimum. As long as the stomates are open and there's plenty of carbon dioxide coming in, Rubisco is connecting with the carbon dioxides. It's fixing the carbon dioxides, and we're all good. But remember, I told you in the last set of notes that sometimes those stomata have to get closed, and they're closed if water loss becomes really great. Water can evaporate out of the stomata, and so the plant sometimes has to close them because it's desiccating, it's losing too much water. So let's see if this one tells us what we need. So it says here, back up. Most plants are called C3 plants. They have no adaptations for minimizing photorespiration. Both stages of photosynthesis are occurring in the same cell at the same time. So both the light reactions and the Calvin cycle are happening simultaneously. Oxygen and carbon dioxide have to be coming and going through those stomates. Sugars are transported to the vascular tissue through transport throughout the plant, and it's all good. So here's where the C3 plants run into problem. There are environments where keeping stomates open leads to desiccation. If it is a very hot and dry environment, lots of water gets evaporated out and the stomates have to close. Closed stomates mean that no oxygen is coming in. So do you see where it says decreasing, and then the brackets mean concentration of? So decreasing concentration of carbon dioxide on the inside, and oxygen starts increasing on the inside because it's being made and it can't escape out of these closed holes. Remember, Rubisco likes oxygen. So if oxygen starts increasing on the inside, Rubisco is going to start glomming onto oxygen, and that's going to lead to photorespiration, which is terrible. It doesn't do what the plant needs it to do. So the plant is trying to open and close these without leading to photorespiration, and that's a terrible problem if these plants are in hot, dry climates. So there have been some adaptations over evolution. The two major adaptations are called the C4 adaptation and the CAM plants. What you need to know, and I'm not going to go into a whole lot of detail, is very simple. For the C4 leaves, you need to understand that the two processes, the light reactions and the Calvin cycle, have been separated into two different types of cells. So these processes are happening in two different types of cells. You need to know that. In the CAM plants, those two processes, the light reactions and the Calvin cycle, are happening at different times of day. One happens during the sunlight, one happens at night when there is no sunlight. And both of those processes, you're just gonna have to trust me for a moment, limit photorespiration. So these are good adaptations for plants that are located in hot, dry climates. So let me give you a few examples. So C4 leaves, carbon fixation occurs in the mesophyll cells. So carbon fixation is happening here. Oxygen, carbon dioxide is incorporated into a 4-carbon, you don't need to know any of this, the 4-bundle sheath cell. Okay, the 4-carbon acid is then transported to bundle sheath cells. So my picture is covering the bundle sheath cell here. So notice that this is where the light-dependent reactions are happening, and then the rest of the Calvin cycle happens over here. Oxygen starts collecting here, but it's not over here. So this separates the cells that have a lot of oxygen from the cells that don't. And so this allows the process to keep happening without oxygen interfering. So all you need to understand is that with the C4 process, the light reactions are separated in different cells from the Calvin cycle. Now for the CAM plants, oops, sorry, I didn't expect that there. The CAM plants, examples of CAM plants are like pineapples, for example, are CAM plants. Sugarcane and corn are examples of C4 plants. So in the CAM plants, it's actually in different times of day. So carbon dioxide is brought in at night. Carbon fixation occurs during the evening when the stomates are open so that water it's not so hot, so they can open and not as much water is lost. So we can bring in lots of carbon dioxide. And then it's stored. And then during the day, those stomates close. But now there's lots of carbon dioxide in the plant so that the rubisco isn't attracted to the carbon dioxide. Isn't attracted to the oxygen. It's attracted just to the carbon dioxide, and that prevents photorespiration. So what do you need to know? Because those are pretty confusing, I think. You need to know C4 plants, they separated photosynthesis into two different cells. In CAM plants, they separated photosynthesis into two different times of day. All right, and then just wrapping this up, what's the importance of photosynthesis? Consider everyone you know, every pet you've ever had, every ancestor in your lineage, they've all been able to exist for the simple fact that photoautotrophs, photolight, auto means self, troph means food, make more food than they need and produce oxygen gas as a waste product. Modern industry is more and more interested in using plants to do all sorts of things, like make biofuels, for instance. So if you look at this is full of plants. These bags are full of plants, and they're using that to make biofuels. All right, cuties, that is the end. These are awesome review questions that you should try to be able to answer. I'm not going to go over them right now because you have been listening to me too much. Let me know if you have questions.